Welcome to Horton Implants Oral Bites with your hosts, Kate Bootman, Will Murphy, and Carl Horton.
Australia, Kate.
Oh, gosh.
You kind of knew it. You kind of knew it. It had to be there. I avoided the hat. So welcome, mate.
Thank you very much. And obviously, as you can hear by my accent, yes. I'm an adopted Australian.
Yeah.
Okay.
My wife's got family over in Melbourne, so she spent a year over there. So we've amassed Australia paraphernalia.
There we go. Oh, that's very good. Yeah. Well, look, I'm collecting passports. This is my third passport.
Brilliant.
Well, everybody out there, hello, welcome. Hope you've had a fantastic week. We're in touching distance for another weekend. You will see I am joined by the normal duo. We have Will Murphy and we have Carl Horton, and we are absolutely thrilled to say that we are joined by Greg Maier from Orthocel, all the way from Australia. So, Gregor, thank you very, very much for joining us today.
That's an absolute pleasure. Very nice to be here.
So we know a little bit about you and about Orthocel and some of the products that you do. What we would like to hear from you is what Orthocel is all about, please.
Great. And again, thank you very much for having me on. So OrthoCell's got quite a unique story. We started our journey as a regenerative medicine company, specializing in cell therapies in the orthopedics field. Our CEO and CSO were involved in the early commercialization of a cell therapy called the Maier technology, where they took bits of cartilage, extracted the chondrocytes, cultured these cells up in the laboratory, and then these were surgically implanted back into the degenerative cartilage injuries with the viewpoint of growing cartilage. And what's unique about that is you need a collagen membrane in order to act as a cell-carrying device in these treatments. Once they had commercialized this product, they left that company and formed OrthOCell. They had a viewpoint of doing a similar type of cell therapy for treating chronic degenerative tendinopathies, because there are very few treatments that are out in the market that actually deal with this degenerative disease. Very happy to say we're the first company in the world that has been able to commercially produce a product that is available in market in Australia. But again, the fact comes into needing a collagen membrane to act as a cell-carrying device for these tenocytes in rotator cuff surgery, in any other ligament and tendon surgery where you might need these cells to be added. So I suppose our journey towards dental took us down this pathway where we had a look through all of the various collagen membranes that were in market, both in an orthopedic sense and in a dental sense. And we couldn't really find a membrane that was fit for purpose. A lot of the membranes were either cross-linked or of the wrong source material or wrong origin. And that set our Chief Scientific Officer, Prof. Minghao Zheng, down this pathway of developing and inventing the CelGro collagen membrane.
This is interesting for me personally, because I've had 2 knee operations on the same knee, and probably over the last however number of years, I've sort of looked into different therapies that are, appropriate for, dare I say, somebody of my age, because you could get to a certain point where, because you've had these operations, you start developing sort of slightly arthritic knee. It was too many years playing rugby and getting my sort of knees torn about. And essentially the operations I've had have been having the meniscus taken out. And again, one of those therapies that I looked at personally was, growing your chondrocytes and putting them back into your knee. But I had no idea that they were stabilized with a membrane. And are these membranes actually sort of tacked over the top of the defects and the repair? How are they applied?
So again, it was a process of discovery throughout the years. The early generation ACI technologies, the membrane, they used a periosteal cover, so they'd harvest some periosteum. They would then suture the periosteum over the defect and inject the cells under. They then moved to loading the cells onto a collagen membrane and suturing it into the defect using a very small 6-0 suture. And then they went through, which is now the case, where you're using a tissue glue, so a tissue glue, and that is then glued into position.
Oh, wow.
But the key thing, as you're pointing out, is, and I think what a lot of people are learning as what should be the gold standard, is you should be looking for a biological intervention first, because that buys you time. So if a biological augment can give you an extra 15 to 20 years, by the time you have your first replacement, if you need that, you're well into advanced stage, and you should hopefully never require a revision replacement. So it's all about buying yourself time for these next treatments.
Oh, absolutely, yeah, because the last thing I want is any form of sort of knee replacement. Hopefully it hasn't got to that stage, but obviously those are time-limited surgeries, aren't they? They probably give you maybe 10 years, 15 tops, and still plenty of complications. And in my head anyway, I'm still quite young and want to be cycling and charging about. So it's really interesting to see how that's developed. And how do the different types of membrane make a difference to these surgeries?
Yeah, absolutely. And an interesting point, because we come from the cell therapy background, and maybe it's probably worthwhile discussing Professor Minghao Zheng. So Prof. Zheng is an MD, he's a PhD, he's a member of the Royal College of Pathology. And because he has a pathology background, he looks at these treatments and these devices from a very different perspective. So a lot of medical companies, let's take dental, for instance. One of the main problems with membranes, in our opinion, and from what people tell us from around the globe, is handling. So if the membrane doesn't handle, or if it's put in position and it deforms, it loses shape, there's issues closing, etc., etc. That's not ideal. So what have most companies gone and done is they've gone and used bovine materials, or they've cut up and cross-linked the raw material to have a membrane that handles better than the market leaders. But the problem comes that when you cut up and cross-link these membranes, or this raw material, to form a membrane, you're actually altering the ultrastructure of that natural collagen. So rightly so, companies are still saying this is collagen. It absolutely is collagen. But what they may not have realized they've done is they've altered the characteristics of that membrane. And so that membrane may stay a lot longer than it should, which hampers bone regeneration. But also you find that by cutting up and cross-linking, you really struggle to get out all of the impurities within that membrane. And the same thing occurs with membranes that are manufactured. You have some bilayer membranes that are really top of the market, but they still contain minute traces of DNA. So the manufacturing process hasn't been able to get these minute traces out. And these traces of DNA are in no way detrimental to the patient. But what happens is you get a mild inflammatory response when they're placed, and this results in that membrane resorbing a little bit quicker than it should, which then gives you that granular bone as opposed to mature cortical bone at re-entry. So it's vitally important that you have the right source material. It's vitally important that within that source material, you're going for the right type of tissue.
Yeah, I mean, that's true. We see that with some of the membranes, that certainly with the cross-linked, you certainly get a stronger tissue response. So you get a much more inflammatory response with the cross-linked ones compared to the non-cross-linked ones. But the handling characteristics, I mean, I've got a sample, so I can't wait to give it a try. I'm looking forward to that. We're waiting for sort of a nice case where we can have a look at it and see how it works. But yeah, so how do you feel that it differs in terms of its handling compared to, say, the other membranes that are on the market when it comes to dental aspects?
Well, let me take a slight step back again. So if you look at when we developed this product, we looked at ovine, bovine, porcine, equine, and human as a raw material. We also looked at synthetic, but that's another story. And within those 5 species, we looked at multiple source types. So if you look at a bovine collagen bundle, for instance, a bovine collagen bundle is much bigger than human collagen bundles. So for instance, if that is your human collagen bundle, this is your bovine collagen bundle, you're asking for these 2 bundles to interact and to regenerate, and that's just not possible. The body recognizes that this big bundle is actually out of place, and that's what potentially creates that mild inflammatory response. So when we looked at it, we found that porcine was the ideal source material, and within that species is where our IP protection comes, is what we determined was the right source from within the porcine material. And the big key to this is our smart manufacturing technology. So we, again, through Professor Zeng's pathology background, we manufacture our products very differently to what existing companies do. So the way we are able to extract all of the residual DNA, the non-collagenous materials, the lipids, the fats, etc., out of this raw material, and by doing so, we don't damage the natural collagen architecture. So we're left with a very pure collagen membrane that has retained its natural collagen ultrastructure and natural collagen architecture. So when you then wet this membrane, it doesn't lose its shape. It doesn't stick to the instruments or your gloves, but it still has the ability to adhere beautifully over your bone regeneration site. It still has the ability to have sutures placed through it, or pins or tacks. So it hasn't lost that integrity or structure, because we haven't had to cut it up, chop, cross-link it, and reconstitute it. So all of these things lead to having a membrane that handles far better than anything else in the market. But on top of that, it gives you an osteoconductive capabilities. So we're showing in our clinical studies that you're actually getting mature bone at a far earlier time point than existing membranes.
That's excellent, isn't it? It's what we want.
Yeah. How long does the membrane last, if you like, in situ? So at what point also can blood vessels be sort of migrating through that and providing angiogenesis for our grafts?
Yeah, absolutely. That happens very quickly. So the membrane lasts for between 4 to 6 months. It's actually really unique. So we've got a saying where we believe that the integration and degradation profile of our membrane is commensurate with the healing process. So as the body heals, the membrane degrades, and by the time the body is healed, the membrane has completely resolved. So there's nothing left behind. We found in some of the histology at 8 to 12 weeks, you're already seeing this osteoid front where the membrane is in position. So in the histology, you can still see the membrane in position. You can see this red line, which is the osteoid, and you can see that the bone is forming. And within the bone that is forming, you see space within the bone. And around the space, again, you see these red lines, which is the osteoid. So it's almost showing that you're getting bidirectional bone growth occurring.
As a result of the membrane, or is the membrane just facilitating that within the biomaterial?
So again, we believe that this is more than a barrier membrane. And when I've spoken to a lot of people, and even in industry, people think that a membrane is there purely to act as a barrier. And while that's very important to keep the soft tissue ingrowth out, we believe that this membrane actually facilitates bone growth. It's osteoconductive in nature. So it's contributing with the bone graft in terms of forming bone. So within the rough side of our membrane, this is where we find cell attachment and cell proliferation occurring. And this is the phase where the tissue remodeling occurs. So it's a bilayer membrane. The smooth side is creating the barrier. It's preventing the soft tissue ingrowth, but it's still semi-permeable that allows bioactive molecules and proteins to penetrate through to nourish the repair site. And the rough side of these well-aligned collagen fibers, that again is where the tissue remodeling occurs. This is where the cells integrate and proliferate.
So what biomaterial beneath the membrane do you recommend? Are you developing your own products, or do you recommend it with other products? Because some products that we use, as part of the manufacturer's instructions, you have to use the membrane and the biomaterial, and to not do that, you can potentially leave yourself on a sticky wicket medico-legally. So we have to make sure that both things can be compatible.
Correct. So when we did our clinical work and our preclinical work, we compared ourselves to BioGuide, and the bone substitute that was used in a lot of the BioGuide studies was BioUs. So in order to do a fair comparison, we compared ourselves with CelGro and BioUs, comparing against BioGuide and BioUs. So that is in both the preclinical and clinical studies. In clinical use, I'm aware of people again using BioUs. I'm aware of people using autologous bone. I'm aware of people using synthetic bone. So all the report backs that we've had have been, and a combination of those as well. And all the feedback that we have had is that it hasn't impeded in any way one or the other. They've all still achieved very, very good mature bone at re-entry, and often at a much quicker time point.
Oh yeah, no, I appreciate that, but I don't know whether you have the litigious culture that we have in our country, where should patients develop, have a complaint about a dentist or a surgeon, the very clever personal injury lawyers and barristers that we have in our country will look through things like manufacturer's instructions, and they'll pick all this sort of thing apart. So, with BioOS and BioGuide, literally, I think manufacturer's instructions, you must use these two together.
Correct.
And if
we don't have that in our instructions for use, no.
Yeah, if you start mixing and matching, I mean, this may or may not happen to anybody, but this is the way our country is going in terms of.
No, I get that. And I'm not recommending one or the other.
I understand that. As I said, our preclinical and clinical work was done combining CelGro and BioOS.
Yeah, you didn't pick a market leader or anything like that. Then you thought you.
Exactly
one of the obscure ones.
It's a good standard to take a reference from, isn't it?
Absolutely, and BioGuide is a phenomenal product. We honestly believe that. When we did this work, Professor Zeng looked at 45 medical devices in market, both dental and orthopedic, and we truly believe that BioGuide was the market leader. The market share shows that as well. But there are some issues with what doctors have reported back to us in terms of handling and granulated bone at times at re-entry. So can't say anything wrong about BioGuide. It's a phenomenal product.
No, yeah.
We've modeled ourselves on that, and we just believe through our manufacturing techniques we get an even better product due to handling and mature bone at an earlier time point.
I suppose the difference is probably in a different viewpoint from where Will's coming from, is that you're focused on your product, which is your membrane product. You're saying your membrane isn't sort of, sorry about the cat, isn't.
No. I was wondering what that was. I thought it was a sound.
Isn't just fixed to one product, one bone substitute. It seems to be working quite nicely with all of them. Do you think the company might look at sort of bone substitutes in the future? Is that maybe where
we absolutely are? Absolutely. There's a pipeline product in play. It's still too early to talk about that, but there absolutely is a pipeline in play.
But and I mean, we talked a little bit off air about how long's the company been going now? How long has it been?
We've been going for roughly 14 years, and 14. I've been with the company for half that time, for the last 7 years.
Yeah. And
Gregor, you were also telling us about some other people that use your products, some of the surgeons and the plastics. Could you touch on that?
Yeah, so that's well with, and as mentioned previously, we developed this product as a cell carrying device, and what we found was really interesting is that when we provided, and it's important to know, we can manufacture CelGro in different shapes, thicknesses, etc. So the dental application is a certain size with certain thicknesses. Our orthopedic range is a lot thicker scaffold in the orthopedic terms, but we also do a very thin scaffold membrane for nerve regeneration. So, for instance, we've put the orthopedic product into an upper limb surgeon in Western Australia's hands, and we were asking him to consider this for tendon repair in the fingers, and he looked at it and he went, this is phenomenal. I think I can do a lot with this in peripheral nerve repair, which then led us on our peripheral nerve pathway. We're about to publish the data from a 20 patient clinical trial where we're showing repair at 12 months compared to the market leading products that show repair out at 24 months. So we're getting a far better outcome at 12 months than these guys are getting at 24 months. And the reason for that is that because of the way this membrane works, because it creates a bioactive chamber around its repair site, this membrane can be glued into position. So there are far fewer sutures, and it's compliable. So there are far fewer sutures being required for the nerve repair. as opposed to what they do conventionally is they have to suture these nerve wraps that are made from bovine or inferior products that don't comply, they don't mold around the nerves, and you have to then pass a number of sutures through these nerves. So you're almost damaging the nerve to repair, to bring it closer. We have a way now of wrapping that nerve with the CelGro, which then creates a bioactive chamber, as I mentioned, and then facilitates a nerve repair at 12 months. And the results have been so good that this surgeon has now gone from treating trauma injury to treating tetraplegics and quadriplegics. So he takes a healthy nerve from above the fracture site. He then plumbs those healthy nerves into the damaged biceps, triceps, and these patients start going from having very limited movement to being able to hold their kids, to hold a mug, to active their wheelchair. It's phenomenal, the results we're getting. And it's not a miracle cure, it's for a set patient group. But I suppose it is a miracle for those people that are getting these benefits.
Yeah,
very much so.
Yeah, and we put this in the hand of plastic surgeons, and they look at it and go, wow, I can see this indication for the product. We give it to orthopedic surgeons, they go, yeah, I can see this. So it's amazing how many applications there potentially can be, and it's all as a result of how we manufacture the raw material. We are able to make this pure collagen membrane that has retained the natural collagen. And when you look at a scanning electron microscopy of CelGro, you see something that's called the 64 nanobanding on the collagen bundles. And that nanobanding is important because it shows how close the raw or our manufacturing product is to human collagen.
I was doing a lecture last Saturday. I should have asked you to come onto the lecture because it was about this time. You'd have finished about 6 o'clock, but we were discussing all of this. This was part of our formative assessment. So I find it fascinating. What I love about what we're talking about as well is the diversity of where your product is going, because what we tend to find, unfortunately, as dentists, and Will may be slightly different, is that we're kind of sometimes blinkered into what we do. So we look at the products and we see them as dental products, but actually there are far many uses, and that's what I loved about. I mean, when I looked at what you're doing, and it just seems absolutely fascinating. So, have you come from I know we know a little bit about the background, but have you come from a background, and we're now coming into dentistry as kind of like another avenue? Are we kind of early or late in the process?
No, I think you first came off the rank. So
right.
the dental pathway was the first pathway we decided to enter.
Yeah.
Let's be honest, dentists are early adopters as well.
Yeah.
You guys, in terms of implants, in terms of bone regeneration, you're way ahead of potentially where some of the other disciplines have been. So it made natural sense. So if you look again at our orthopedic background, if we can do GBR in dental, there's definitely room in the long bone for these traumatic injuries where you're getting non-union. So that's potentially another pipeline that we're looking to enter into using CelGro as a periosteal cover. You know, there are all these potentials because of the great results that we're getting in dental, with the great results we're now getting in nerve. We've done some rotator cuff work that we're looking to publish, where CelGro is used as an augment, as a patch. A lot of surgeons now in Australia are actually combining cells and the scaffold in their rotator cuff surgery. So there's numerous applications. And as we discussed right in the beginning, the ACI technology, which was 20 years of data now, out in the market, you know, that's a proven technology, and we're able to do that now in Australia using CelGro with the chondrocytes.
It's fantastic. You mentioned a little bit about synthetic, and you said that's another story. Did you explore the idea of producing a synthetic membrane, or are you you might not be able to answer the question?
No, we did. We absolutely did explore that, and I think we decided, you know, very early on that wasn't the right pathway to go.
Yeah.
There's massive inflammation. Look, I think there's definitely room for some form of synthetic material in certain indications, and we're actually looking again from a pipeline perspective at creating a collagen membrane that potentially has reinforcements within it that can look at that, can look after that market. Professor Zeng is a wonderful innovator. One of his strengths is what is collaboration. He doesn't keep everything to himself. He believes in discussing these things with as many people as possible to get the right outcomes for the patient. Yes, we did look at synthetic. We didn't believe it was the right pathway to go.
I think you've hit the nail on the head in terms of stability, because I think with any membrane and GBR, one of the things that you probably can't always guarantee or know how effective the stability of the whole graft is. You know, we're putting in a granule material, we're laying over some bits of paper, and we're sending our patients off into the big wide world. Now, I know many of us, probably just out of instinct rather than any clear direction, do double layers. Do you do a double layer on your membranes? Have you got any guidelines for that, or are you talking about innovation to provide extra rigidity?
So we don't. So in terms of doing a double layer, we don't prescribe that. I do know that, for instance, Nick Fahey does do a double layer at times, but that's his preference, and he freely chats about that, saying that he's got some extra membrane, and he then, in some instances, he'll do a double layer. From a rigidity perspective, we have people doing vertical ridge augmentations with the CelGro membrane, and then they're tucking in his position, and they're creating this beautiful ridge with the bone substitute and the membrane holding it in place. Again, it's not going to be an indicator for every single vertical ridge, but it is being used, and a lot of people are now using it as well in combination with a synthetic membrane, so that they're getting the bone substitute with the synthetic membrane with the bone substitute CelGro, the synthetic membrane over the top of that. But from a pipeline perspective, we're hoping to come up with something that is resorbable, so the whole construct will be resorbable. We also, from a pipeline perspective, looking at silver nanoparticles, so that the membrane is loaded with silver nanoparticles for infection control. So that's we've published that paper, so you'll be able to pick that up on PubMed.
Yeah, I mean that's another thing that needs solving, because every now and again, sort of loose granules or spaces within a GBR can be a perfect culture medium, and you know, every now and again you get these little infection bubbles inside your GBR that can be pretty annoying. So solving that problem is going to be very useful.
Yeah. Prof. Singh's made some phenomenal headway on that, on silver nanoparticle membrane. So, you know, hopefully it's not too long, but these things do take time to get it right. Again, Prof. Singh believes in that translational, as do we, the translational medicine process, from proof of concept through your preclinical to clinical and then into the clinic. So we don't believe in taking shortcuts. It's got to be done the right way.
Yeah.
And you see, for that reason, Will, it's why I'm going to speak to Gregor off air, and I'm going to sign you up for a clinical trial for your knees. We'll get you sorted.
I'll very happily be a guinea pig. I'm good for that. And actually, my son he's a professional rugby player. He's just done his shoulder in, so we might be needing a few truckloads of.
What's quite funny, and this is not a good advert for our company. It's we have one of our sales team who's an ex-Wallaby rugby player. I played international hockey for South Africa. We had Paul, our CEO, who played highly competitive cricket, and we all at that stage where we're too far gone for biologics. So we all at a congress, and we all limping. And the three of us are limping down there.
Yeah, I know the feeling. Yeah.
And unfortunately, I think we missed the boat.
Yeah, probably. Yeah, I think the career's over. I might as well hang up my boots. I'm done.
There was a successful story for one of the Australian footballers, I believe, Gregor. He had some work done and had some tissue repair, I believe.
Yep, that was with our autologous tenocytes. So that was we call it OrthoATTI. So it's autologous tenocyte implantation. So this is where patients have failed conservative treatment methods for tendon injuries. So a good way to explain it is, if you've got a tennis elbow and you've had that for a long period of time, you've tried to rest it, you've tried physiotherapy, you may have tried a corticosteroid, you may have tried platelet-rich plasma, you may have tried shockwave, and what you find is that your symptoms may resolve for a couple of months, but they come back. And that's because you're not treating the underlying pathology. And in the early stage of tendinopathy, those treatments are fantastic because it takes the pain away. You can load the tendon, and by loading the tendon, it helps restructure the tendon. But when you get to that chronic degenerative stage, you get cell death occurring within the tendon. And what happens is the tendon goes from this beautiful structure to just completely breaking up. You have fatty infiltrations, you have angiofibrotic hyperplasia occurring. It just becomes a mess in that late stage tendinopathy. And there are no real treatments for that. So what Prof. Zink hypothesized was, well, if we take cells and we can put cells back into the area of cell death, what will happen. And through that translational process, they looked at mesenchymal cells, they looked at adipose cells, they looked at bone marrow aspirate. And in conjunction with the regulators said you need to have an autologous and homologous cell, which is why they were then back to tenocytes. And no one else has been able to do it because it's so difficult to do this in a commercially viable way. Prof. Zeng has found a way. So a small tendon biopsy is taken in an outpatient setting. That then comes back to our GMP lab, where we isolate the tenocytes, culture those cells up to a significant volume, and those cells are then re-injected back into the damaged tendon under ultrasound, so into that hypoechoic area of the tendon. And what happens is these tendon cells then engraft. So it's a patient's own tendon cells. They engraft into the tendon. They start synthesizing and laying down a collagen matrix. And that's what then helps restructure the tendon from that mess to back into a normal tendon or normal-like tendon under load. So Prof. Zeng has also worked out the exact amount of load that you need to apply. So he's actually grown human tendon in a bioactive chamber in the lab by working on that. You need 6% of load on this tendon. So he's taken this tendons in a petri dish. They form a layer at the top. He's rolled them up. He calls it a spring roll. He's put them in the bioreactive chamber. He's applied load, and he's grown human tendon. And where this is brilliant is that means that one day for an ACL repair, you may not need to harvest your hamstrings anymore. You may just need a small tendon biopsy and grow your own tendons. So again, that's blue sky. That's pipeline, way down the line. But the early lab work that Prof. Zeng has done, and to be able to grow human tendons in the laboratory, validates our tendon cell therapy, to shows what happens in the body.
Yeah.
So it's really exciting.
Oh, it is, isn't it? I mean, this is just exciting. You know, I mean, we're talking now, and I'm ready to do a graft. It just makes me want to get out there and work with the things and help people out. I don't know about you, Will. Well, maybe I'm strange on that.
I look forward to two things from the three of you. Is once when you've used the product, and then at that 4-month mark when you go back to re-entry and you see the quality of the bone. That is what I'm really looking forward to hearing from the three of you. And perhaps you can let me know in that 4-month time once you've used it, how it handled and the quality of bone you've achieved.
Yeah.
What we're also missing as well, Gregor, I mean, for people out there that are wanting to try this product, I mean, I know where we can get it from, but who's distributing it over in this country?
Absolutely. In the UK, General Medical is just distributing the product for us. So I'm sure we can share the link to their site.
Yeah.
That will be great. But John at General Medical is doing a great job.
Brilliant.
He is indeed. I can see these two twitching. They're desperate to go and use it now.
I can't wait as well.
Only on teeth, though. I think.
No, you want to be the next patient. I'll be
the knee patient. Yeah.
I'll do it for you, Will.
Oh, thanks very much.
There we go.
We'll get you the handbook.
Yeah.
Sign him up.
The old Haynes manuals. I'll just be doing that. It can't be much different, can it? It's got to be.
How hard can it be?
I'm used to 6 zeros.
YouTube it.
Yeah. That's what we do anyway.
There we go.
Yeah, we do that all the time. So, we run out of time.
No, you carry on. No, I was going to ask Gregor. Carry on.
In terms of sort of where do you think? What's your next big thing? Do you think that, if you're allowed to talk about it, where's the sort of the next step with you guys? Do you think what's coming out?
Well, from a dental perspective, we recently achieved our FDA clearance. We've just had our first few cases done in the US by some of the key opinion leaders that are working with us. So that's incredibly exciting. We're looking forward to doing a full launch there in the not too distant future. We've obviously had our CE mark for the UK and Europe for the last 18 to 24 months. So that's phenomenal. And we've just recently received our Australian regulatory approval for CelGro as well. So 3 dental markets that we are now in, the 3 main jurisdictions for ourselves. So that's very, very exciting. From a further company perspective, we have our nerve product. We're hoping to have Australian approval in the not too distant future, and we're looking at a breakthrough technology approval in the US. So that's something that's really, really exciting.
And I guess
I can say that because that's just been released. So that's public knowledge, being a public listed company.
So I guess all the different, like Europe and Australia and America, have different kind of regulatory issues that you guys have got to step over. and you probably find one is easier than the other, but they're all probably quite just as difficult.
Absolutely.
Yeah.
Absolutely.
Yeah.
Yes, it's all unique in their own way.
Yeah. Wonderful.
I'd imagine there's a large portion of time spent by a team over there that pull their hair out at times, I'd imagine.
Again, we're very unique as a company. A lot of the people that work for us have a science background. They're either production scientists, researchers, etc. But we also have a regulatory department that works and looks after that. So that's a massive task in its own for our regulatory department team.
I should think so.
And they again work hand in hand with our clinical research team. So there's a lot of work going on in the background to make this possible.
Wow. So you're in Western Australia, is that right, Gregor?
That's correct. So we in Perth in Western Australia. And it's interesting, I was in Perth in Scotland just before COVID struck. So it was 30 odd degrees Celsius close to 40 in Western Australia in Perth. And while I was in Perth in Scotland, it was, I think, -5 or something.
Yeah. But that was on a good day, probably.
Yeah, midsummer. Yeah, midsummer.
But
Midsummer.
being what Western Australia is phenomenal for is being an incubator market for all these regenerative medicine technologies. There's some wonderful innovation coming out of Western Australia, not just in what we're doing, but there are a number of companies in this space. And you can almost find the right pathways to commercialize products within this market and then take it on to different jurisdictions. So although probably the most isolated city in the world with a population of over 2 million people, we're a phenomenal market and there's amazing stuff happening in the regenerative space in Western Australia. And as you can hear, my Australian accent is very strong. So for a South African born to be saying that, you've got to know it's good.
Yeah,
absolutely. Gregor, I mean, you've given us some background on the company, which is absolutely fantastic. I am interested in your background, especially with your Australian accent. Where have you come from? How has it led you to where you are now?
So I started late. As I mentioned earlier, I played international hockey for South Africa. So that took up a long period of my time. No real movement for full-time work. I lived in the UK for a long period of time as well, and got into the pharmaceutical industry. On moving back to South Africa to get married, I again got into the medical devices industry. After a long period there, I was offered an opportunity in Malaysia in the non-surgical field. So that was really interesting. And then back to South Africa, into the digital operating theatre space. With a company called Carl Storz, and then I found my way to Australia and into regenerative medicine. So it's been a steady pathway from pharmaceuticals through to biologics.
Wow.
Wow. See, I usually just say I went from Cardiff to Birmingham, and that's the end of my brief. A
bit of a nomadic family, I think.
Wow. So in the UK, where were you based in the UK?
Again, in wherever I was able to play. So I was in Fulham and Wimbledon Village for a period of time. I lived in Oxford, in a little village called Woodstock, just out of Oxford, for a period of time. I lived in Northern Ireland in Belfast for 2 years.
Oh.
And then ended up in Wimbledon Village in the last couple of years in the UK.
Oh, fantastic!
All great places, all really good places. Great places, great people.
Yeah. So, do you have a favourite? Did you have a favourite?
In terms of the UK?
Yeah.
You got to love London. I love London. What a great place.
Fulham's nice.
Yeah, Fulham's great. But yeah, I love London. The diversity in London, the history, it's just superb. I mean, Oxford was great as well.
Oh, Oxford, yeah, very nice. Do you miss your hockey?
Yep, I do. But as I said, I battle to walk after mowing the lawn.
Yeah.
So it's I played the odd vets game, but I've got children now that are playing some sports. So I've taken a back seat so that I can watch them play. I found that last year I missed a couple of games, and the fact that I'm not traveling as much, it's great to be watching the kids play a bit. Yeah,
oh, I'm sure. And Will, you just said earlier, that's something I actually didn't know about your son playing rugby.
Yeah, well, my two both play. The youngest just went through the academy pathway with Worcester Warriors, who are firmly rooted to the bottom of the Premiership table, unfortunately.
They're the strongest, the ones at the bottom.
Well,
they hold them all up.
Yeah, they're resilient. Fortunately, there's no relegation this year, so they get another stab at the Premiership next year. So he's academy, so he's trying to sort of work his way in and develop himself. But yeah, he just phoned me up yesterday telling me his shoulder's killing, and I think he's having a scan done today. So
the Tina sites could work.
There you go. I'll be referring him on.
There's two people for you, Gregor. I'll tell you now.
Oh, geez, this is great. Yeah. When I lived in the UK, I was supporting Saracens. I'm sorry to say.
Oh well.
And then in Belfast, it was Ulster. So I had to support the local side in Belfast.
Both good teams.
Yep.
Was it Fulham when you were in Fulham as well as a football team, or was that not
Chelsea?
No, unfortunately, I was Man United. From so Gary Bailey used to be the goalkeeper of Man United back in the 80s, 70s, 80s.
Yeah.
And as a South African in the UK, or as a South African playing in the UK, they came out to South Africa in the I think it was early apartheid days as well, and I still remember that. So I've got a knitted jersey, Man United. So. But I'm not as passionate as my brother-in-law, who's an avid Liverpool fan.
Yeah.
Okay. Very good.
Oh, very good. Well, gentlemen, I hate to tell you, but we have reached that magical time today. So I would like to say a massive thank you, Gregor.
Huge.
I mean, massive. If we could entice you back, we would love it if you would come back.
Absolutely.
That would
it would only be a pleasure, and it's wonderful meeting the three of you. And as I said again, I'm really looking forward to your feedback.
So,
we'll
absolutely.
Definitely. Well, let's do this again. That would be a lovely chance to share some feedback with you. So
that sounds great.
That would be wonderful. Will Murphy, as always, thank you very much, kind sir.
Welcome.
Cole Horton, thank you very much.
Yeah, and thank you very much.
Everybody out there, I hope you've enjoyed it as much as we have. I mean, we would love to keep Gregor now, but we know this gentleman.
Stop. I want to carry on.
I know.
I've got to go and do some teaching.
This gentleman needs to go and start his weekend. So, Gregor, honestly, thank you again. It's been absolutely wonderful.
Yeah.
Have a great weekend, and have a great weekend, everyone.
Yeah. Everybody, have a lovely weekend.
Cheers. Thanks a lot.
Take care. Bye-bye.
Bye-bye.